So welcome everyone to our IMPART Research Reels webinar. This is a new webinar series that IMPART is hosting. Before we get started, I would like to introduce it to you all. So IMPART Investigator Team is a national virtual research institute headquartered at Dalhousie Medicine, New Brunswick in St. John. Our team is composed of 45 academic and clinical partners across 8 universities in 6 provinces, as well 25 community and funding agency members, 15 private sector company representatives, and 2 provincial government departments. IMPART's mission is to think boldly and utilize our network to improve the outcomes of vulnerable patient populations, starting always with innovation developed and tested in New Brunswick. We apply intersectional thinking between social and medical innovation and have brought together a diverse and inclusive team of experts to support activities from fundamental research to applied interventions with vulnerable populations. So the Research Reels webinar series will host monthly webinars by IMPART team investigators and trainees on ongoing or completed research within the network. The objectives of this webinar series are to improve networking among IMPART team members, enhance opportunities for future collaborations, and support knowledge translation of research findings. So if you'd like to nominate an upcoming speaker, please do so in our post-webinar survey. You can also find us on social media platforms, so please follow us to find out more about the research collaboration opportunities we have and more. I am beyond excited to introduce to you today's speaker, who is Erica Seelemann. Erica is a Master of Science candidate under the supervision of Dr. Ketul Chaudhary at Dalhousie University in the Department of Physiology and Biophysics. She is investigating sex differences in right heart disease. Erica has concurrently completed all components of her certificate in science, leadership, and communication. Before her master's degree, Erica completed a bachelor's of science at Dalhousie with a major in neuroscience and a minor applied ethics. Now I'll pass on the floor to the speaker itself.
Thank you for the introduction. Let me just share my slides. Can you see my presentation?
Yes, we can.
Amazing. So thank you all for joining me today, and thank you to IMPART for inviting me to speak. Today, I will be discussing my work, which is focused on understanding how adaptations in the right side of the heart differ in males and females. And just as Candace said, I'm at Dalhousie University under the supervision of Dr. Ketul Chaudhary, and overall my lab is focused on sex differences in right heart disease. Before I dive into the science, I would like to highlight why we have examined sex differences and how it's necessary that we examine the role of sex in research. Health research often focuses on males, and the absence of females in treatment development often leads to unexpected responses when we treat females with the treatments that were devised for males. Females also have more adverse drug reactions than males because of that. There are also unknown sex differences in disease, and they're impossible to identify without explicitly examining the role of sex. This is an issue that extends into heart disease research and treatment. The Heart and Stroke Foundation has identified that 2/3 of all heart disease and stroke clinical research is focused on men, and they have prioritized research that includes females. Studies investigating all sexes are important to make sure that everyone can receive the best possible care. Our lab is specifically focused on how the right ventricle works. The right ventricle is the bottom right chamber of the heart that's responsible for pumping deoxygenated blood to the lungs so that it can become oxygenated. This is a low pressure, low resistance system, and it's very different than the left side of the heart. The left side of the heart is responsible for pumping blood to the entire body. It's a much higher pressure and resistance system, and it has also been the major focus of heart research for several decades, while the right side of the heart has been largely ignored, resulting in a lack of specific treatments for the right ventricle. There is an assumption that the right side of the heart could be treated using therapies designed for the left side of the heart. This is not the case, and it may be because the right and left sides of the heart have very different roles during fetal development, different thicknesses, different masses, and different pressure and resistance environments. Left heart treatments are ineffective on the right side, and there is little support doctors can offer when the right side of a patient's heart is failing. So to reiterate, research on the RV is needed to fill a gap in medicine because there are currently no treatments available for right heart failure. Right heart failure is what our lab is very specifically focused on. It's a condition in which the right ventricle is unable to push a normal amount of blood into pulmonary circulation and meet the body's demands. Right heart failure develops as a consequence of many other diseases in the body. Moreover, research has shown that our right ventricular function is a very strong predictor of survival, as shown by these figures on the right. The figure on the top shows that patients with stronger and consistent RV function have much better survival rates than those who do not. The figure on the bottom shows that one of the strongest predictors of survival is stable RV function. Patients with declining RV function have much poorer outcomes. It follows that if you can stabilize RV function, then you can preserve survival. However, we do not have right-sided specific therapies, and studies have shown that therapies used for left-sided heart failure are ineffective on the right side. Given the importance of RV function for patient survival, it is necessary that we develop specific treatments for right heart disease to mitigate the current deficit. That development requires a stronger understanding of the mechanisms behind right heart failure, which I have examined and will share today. Before I do, it's important to mention that there are major sex differences in RV function in the healthy state and in right heart disease. In our healthy state, females have better RV function than males. And this translates into females having better survival outcomes in right heart diseases. The problem is that nobody knows why these differences exist because of the lack of research examining females and the right side of the heart. And these problems bring me to my questions. Functionally, and at a cellular level, what happens when the right side of the heart is subjected to an increase in stress and pressure? And what, if any, differences exist between males and females in these adaptations? In our study, we use the pulmonary artery banding method on adult rats. PAB involves surgically constricting the pulmonary artery, which carries blood from the right ventricle to the lungs to become oxygenated. And this band limits how much blood can pass through. This causes a pressure overload in the right ventricle. We confirm the success of using PAB in our experiment with right ventricular hypertrophy and right ventricular systolic pressure. Right ventricular hypertrophy is a measure of the heart size. It increases in response to a pressure increase. So you would expect that when we restrict the pulmonary artery, the right ventricle would increase as it adapts to the increase in pressure. And that's what we see here. In males and females, there was both an increase in right ventricular hypertrophy. We also see an increase in right ventricular systolic pressure. Right ventricular systolic pressure is the heart's pressure at the peak of its contraction. It's the highest pressure, overall pressure, that we would see. These measures show significant increase in hypertrophy and pressure. We also saw an increase in RVH at the 2-week time point in males and females compared to baseline. And again, these were similar when we looked at RVSP. Next, we assessed RV function and structural changes using echocardiography. Functionally, data shows that overall measures of heart function were superior in females when compared to males. That's what we see here when we see females and males, and in fractional area change, which is again a measure of overall function. Females maintained function, while in males, it was significantly reduced. Additionally, RV internal diameter and cardiomyocyte size were increased in males at 2 weeks post-PAB. Just like how right ventricular hypertrophy is a measure of heart size, The RV's internal diameter measures how big the inside of the RV chamber is. The following measures show changes in heart size here and at a cellular level. Here we see a measure of the size of specific cardiomyocyte or heart muscle cells. The increase in hypertrophy and preserved function that we've seen in females is typical of positive or effective remodeling. In males, we see an increase that is not concurrent with maintained function, and that's typical of poorer adaptations to disease. Once we knew that the PAB induction successfully induced stress, we wanted to examine what happens more at a cellular level. Previous research has shown that adaptations involve changes in blood vessels. Specifically, hearts that adapt well to stress see an increase in the number of blood vessels proportionate to the increase in heart cell size. And that's what we see here. Each of these pink dots represents a capillary, or a very small blood vessel, and the hexagons represent cardiomyocytes, or heart muscle cells. Moving from the normal right ventricle into a positively or a strong adaptation to disease, we see that the number of blood vessels increases as the size of the cardiomyocytes increase. In a poor adaptation or in a maladaptation, we see that we do not see a proportionate increase of blood vessels concurrent with an increase in cardiomyocyte size. The lack of blood vessels is unable to adequately supply the cardiomyocytes in a poorer case, and this leads to right heart failure. Knowing this, we began our investigation with genetic markers that are responsible for increases in blood vessels. These figures compare the expression of pro-blood vessel genes in the right ventricle of male, here, and female rats, here and here, subjected to PAB. PAB is the intervention on the right. In response to stress, we see an increase in these genes in the female right ventricle that is not seen in male rats. When you compare here and here, this expression is not the same in both sexes. This data indicates a stronger blood vessel response in the female RV here compared to the male RV. To validate what our gene analysis found, we've seen sections of rat hearts with factors that identify endothelial cells. Endothelial cells are the cells that make up the inner lining of blood vessels. We counted these cells in the right ventricle. Our data shows that at the 2-week time point, there's a trend towards an increase in the number of endothelial cells in the female PAB condition compared to the male PAB condition. The work is ongoing, and we're adding more samples to all of our groups. With this in mind, these early findings further support that females have a stronger blood vessel response than males. You can visualize what's shown on the graph here on the left with the arrows. Each of these darker points here, indicate cells that form, again, endothelial cells, the inner lining of blood vessels. In summary, right heart failure is understudied, and there are currently no effective treatments. There is an urgent need for new therapies to fill this clinical gap in medicine. Secondly, there are major sex differences that exist in the right side of the heart in a healthy state and in a disease state. Understanding these differences may aid in the development of new, effective, specific treatments. Finally, females demonstrate a stronger ability to effectively adapt compared to males when there's an increase in pressure by developing new blood vessels. And taken together, these findings are the first step towards developing these therapies for right heart failure, with a focus on blood vessel growth and development. To conclude, I would like to acknowledge my supervisor, Dr. Ketul Chaudhary, for all his support, my committee, Dr. Quinn, Dr. Howlett, and Dr. Dong, as well as my lab members, Sheila Panchakshari, Tony Al-Rabahi, and Kirshani Kesavan, our collaborators, Dr. Dang and Dr. Stewart, as well as Dalhousie University and Research Nova Scotia for funding the project. And finally, I would like to offer a special thanks for the animals that I used in this study. Thank you, and that concludes my presentation.